Hey everyone, Carlson here to cover the beginning of Unit 8 with you, which is the cardiovascular system, and there are 3 chapters included in this unit, and we're going to start off with Chapter 11, which goes over your blood. Now, the living body is in constant chemical communication with its environment, and even though these chemical exchanges are occurring at specialized sites, they affect every cell, tissue, and organ in a matter of moments because all the parts are connected with the cardiovascular system, which is kind of like a car cooling system where our blood is like the water or circulating fluid. Our heart is like the water pump, and our blood vessels are like the flexible tubing that basically exchanges this fluid. Now, blood has several important functions and unique physical characteristics. It is a specialized connective tissue containing cells suspended in a fluid matrix. There are 5 major functions: transportation of dissolved gases, nutrients, hormones, and metabolic waste, regulates the pH and ion composition, it restricts fluid loss, defends against toxins and pathogens, and stabilizes body temperature. To get in a little more depth about a few of these, transportation would involve things like carrying oxygen from lungs to tissues, carbon dioxide from tissues to lungs, distribution of nutrients absorbed at the digestive tract, carrying hormones from endocrine glands to target cells, and absorption and transport of waste to be taken to kidneys and then excreted through our urine. Restriction of fluid loss is due to the ability of blood to form a blood clot at a site where a vessel might break. And stabilizing body temperature, it occurs by absorbing heat generated by skeletal muscles and then redistributing that to other tissues, like we talked about with the skin. The surface will look reddish because those capillaries are bringing blood to the surface to be radiated out to help with that heat loss. Composition of blood, it's made up of plasma and formed elements such as blood cells and fragments. We can separate them through fractionation. We usually do this for analytical purposes. And whole blood from any source, whether it's venous, capillary, or arterial, has the same composition. 100.4 degrees Fahrenheit is the average temperature. Viscosity is about 5 times more than water, and the pH is slightly alkaline, between 7.35 and 7.45. The way we collect blood and analyze it is by venipuncture, which is a piercing of a needle through the superficial, usually median cubital vein, because that's usually the biggest one and most obvious one for many people. Venous blood is most commonly analyzed because there's lower pressure and there's quick healing abilities at that point. Arterial blood tends to bleed a lot more and be more difficult to heal, but we do use arterial sticks when we're evaluating gas exchange efficiency at the lungs, for example. Besides just doing some testing with the blood as far as on the chemical level, we can also do a blood smear with these collections, and that just requires 1 drop. You fix it and stain it, and you can count the amount of red blood cells and white blood cells and lots of other things. Now, we can also get a drop of blood from the finger, earlobe, or toe and heel, most commonly used with infants. 11.2 talks about the plasma, which is the fluid portion of the blood. Plasma and interstitial fluid account for most of the volume of the extracellular fluid of the body. Plasma makes up the greatest volume of whole blood and includes proteins, solutes, and 92% water. There are 3 primary proteins, albumin, globulins, and fibrinogen. And because the liver synthesizes 90% of plasma proteins, liver disorders can cause problems with blood composition. Albumin is the most abundant plasma protein and helps maintain osmotic pressure. Globulins are the second most abundant and contain antibodies and transport proteins. Together, they also help transport insoluble lipids, and globulins that are involved in this are called lipoproteins. Finally, fibrinogen functions in blood clotting. They form fibrin, which is the framework for a blood clot. If we were to take samples of blood that we don't prevent from clotting intentionally, usually, fibrin will form. That plasma is now called serum, and different testing can be done on serum than can be done on plasma. Our red blood cells are covered in 11.3, and they are formed by erythropoiesis. They are the most abundant blood cells, and a red blood cell count is a common test done. It's the number of RBCs per microliter of whole blood. For males, that's 5.4 million, while females have 4.8 million. 1/3 of the 75 trillion cells in the human body are red blood cells. Another test that's commonly done is a hematocrit. It's the percentage of whole blood versus formed elements, and both of these tests can give us some indication of many different diagnoses. The structure of the red blood cells are specialized to transport gases. They are biconcave discs with a thin central region and thick outer margins. They give RBCs relatively large surface area to volume ratio to increase diffusion rate. They are also very flexible and able to bend to fit through those small capillaries and other tubing. During formation, RBCs are going to lose many of their organelles, including ribosomes, mitochondria, and nuclei. So therefore, they cannot divide. They do obtain nutrients from surrounding plasma, and this ensures oxygen's absorbed will be carried to peripheral tissues. And here's a couple pictures. This is of a stained smear of red blood cells. Here is a colorized electromicrograph, and this is just a diagram showing you the biconcaveness of the red blood cell. Now, a mature RBC consists of a cell membrane enclosing a mass of transport proteins, and a molecule called hemoglobin accounts for over 95% of an RBC's intracellular proteins. So they're very important for the red blood cell. They're responsible for the cell's ability to transport gases. There are two pairs of globular proteins that combine to form a single molecule of hemoglobin. One is the pigment heme, which binds iron to allow for oxygen interaction. Bound oxygen equals bright red blood, while lack of oxygen is going to give us a burgundy or really dark blood. If we have anemia, this is a lack of oxygen carrying capacity of red blood cells, and that's one of the things that can be identified through a low hematocrit test. Now, the lifespan and circulation of a red blood cell is about 120 days. That's because they're exposed to several physical stresses. One round trip from the heart through the peripheral tissues and back to the heart takes less than a minute. This lifespan, or this exchange of blood, normally goes unnoticed because they're constantly being replaced under normal conditions. Now, we can also recycle our hemoglobin, and as red blood cells age or are damaged, some of them rupture. And so this results in hemoglobin breakdown in the blood, which can be filtered through the kidneys and excreted by the urine, which would be called hemoglobinuria, and gives you a reddish-brown urine. Usually this can be an indication of different problems. Now, only 10% of red blood cells survive long enough to rupture or hemolyze. The rest are broken down by phagocytic macrophages, or some of our white blood cells that recycle the parts for us. The heme is stripped of iron, converting to biliverdin, which is a greenish color usually that you see when you bruise. The biliverdin then converts to bilirubin that is released to the bloodstream. When this builds up, we can see what is called jaundice, or an orangish yellowish condition in the skin. But our body normally would absorb that and take care of that on its own. Iron binds to transferrin then, which is a plasma transport protein, to continue synthesis of hemoglobin, and again, so that the red blood cells can utilize it to bind iron and therefore carry oxygen again. Now, I mentioned earlier that RBCs are formed through erythropoiesis, which is the formation of red blood cells, and it occurs only in red bone marrow or myeloid tissue after embryonic development. This tissue is located in the vertebrae, sternum, ribs, scapulae, pelvis, and proximal limb bones. Other marrow areas containing fatty tissue called yellow marrow, and during severe stresses like extreme blood loss, that this can convert yellow marrow to red marrow to add in the red blood cell production to make it that basically to help fight that blood loss. Finally, origins and differentiation of red blood cells, platelets, and white blood cells can be seen in this diagram. This side over here, these myeloid stem cells, bring us over to the branch of red blood cell maturation. And this is something we'll cover a little bit more in class. Later on, we will also cover the maturation of different white blood cells that are shown over in this region. So we'll cover this a little bit more in class. And the last thing I just want to mention is that an adequate supply of amino acids, iron, and vitamins are necessary for protein synthesis. And without vitamin B12, normal cell divisions cannot occur. So we have to have these essential supplies in order for this maturation of red blood cells to occur. All right, I'll see you guys next time.